All right, I think we get started. I'm really excited today because we have Dr. Rose Cory as our seminar speaker. I've met her a couple of times on panels, and I'm really excited about her work. So Dr. Rose Cory is a professor at the University of Michigan and an expert in geochemistry and aquatic carbon cycling. And she comes to us from the University of Michigan from the Department of Earth and Environmental Sciences. And she received her PhD from the University of Colorado in Boulder, and then did a couple of postdocs at the University of Minnesota and at Los Alamos. And then she became faculty at North Carolina and then moved to Michigan. And she's really a rising star, or already shining star, in the field of aquatic carbon cycling. And today she will talk about Arctic amplification of global warming strengthened by sunlight oxidation of permafrost carbon to CO2. And I'd like to welcome her to the ESS department. And thank you all for coming to the seminar.
Thank you, Claudia, for that really nice introduction, for inviting me and for hosting. And thank you all for inviting me. It's a real pleasure to get to talk to you about my research. So I assume everyone can hear me, or someone will let me know if they can't. So I'll start by acknowledging my co-authors and collaborators on the work I'm going to tell you about today, including my current and former students, Colin Ward, Jenny Bowen, Emma Reeve, Adriana Trusiak, as well as my current and former lab technicians, Katie Pollock, Leah Treybergs, and Jason Dobkowski, and also my co-PIs on this projects, George Kling and Byron Crump. So I want to set the stage by defining what I mean when I say carbon cycling. So let's go here. Yeah. So carbon cycling, when I say that, what I mean is the study of how carbon changes form from inorganic carbon to organic carbon, and how these different forms of carbon move between pools, such as between the atmosphere and the biosphere. For example, when plants take up carbon dioxide from the atmosphere and convert that to biomass or organic carbon that, as it's decayed through respiration, is returned back to the atmosphere as carbon dioxide. Now, on land anywhere, but particularly in the Arctic, you can't close this loop or this carbon budget without considering that some of this soil carbon is leached into water where it flows into surface waters. So as water moves through soil, it dissolves some organic carbon, and whatever dissolved is called dissolved organic carbon, or DOC, as I will talk about today, or use the term DOC today. DOC flows from land into fresh waters, where it can also be returned to the atmosphere as carbon dioxide, both by microbial respiration, just like that as happens in soils, but in surface waters, we also have to consider that we have sunlight. Sunlight can also oxidize dissolved organic carbon and turn it into carbon dioxide by a process called photomineralization. Right, so there, where you are in sunny California, I think probably many of you are familiar with the process of sunlight oxidation. So, for example, if you wear a colored shirt in the sun often enough, eventually that color will bleach or fade with sunlight exposure. What's happening is that there's some dye that's made usually of an organic compound that absorbs light, hence its color. And as it absorbs sunlight, reactions are initiated that break down that organic compound and turn it into some less colored product. And just like with clothing or with paint, we can detect the sunlight oxidation of DOC even using a smartphone camera, which is shown here in this series of photos of water containing DOC exposed to sunlight over time. What you can see is this dissolved organic carbon, the color goes away. And what we're interested in is what happens to those colored molecules. And what's happening is that as the sunlight absorbing fraction of DOC, which is called colored DOC or chromophoric DOC, is absorbing sunlight, and in the process, some of that DOC is oxidized completely to carbon dioxide, and some of it is chemically altered or changed in its composition. And together, that complete and partial oxidation of DOC leaves behind DOC that is bleached or less colored compared to the original DOC. And together, so if we look at this complete oxidation of DOC to CO2 and the, which we call photomineralization, again, because DOC is turned into carbon in inorganic form or mineral form of carbon. And then we also consider both, we measure the partial photooxidation of DOC. So that's any DOC that's oxidized by sunlight, but isn't oxidized all the way to carbon dioxide. When we look at both of those processes together, they account for the majority of DOC processing in the water column of streams and lakes in the Arctic, which is what is shown here in this pie diagram. So of the DOC that is processed in Arctic freshwaters, about half of the CO2 that is produced is due to bacterial respiration. So, you know, if we think about the total CO2 produced from DOC processing the water column, bacteria contribute about, through respiration, about 25%. And then sunlight oxidizes DOC to CO2, again, process called photomineralization, and produces about as much CO2 as bacterial respiration of the DOC. Then in addition, if we account for that partial photooxidation of DOC, that accounts for about 50% of the DOC processing or degradation of DOC in the water column. So if we add up the partial and complete oxidation of DOC by sunlight, that accounts for about 75% of the total DOC processing in the water column. Meaning if we only look at the more conventionally measured microbial respiration of DOC, we'll get the wrong answer about the controls on DOC processing, as well as how much DOC is processed in the water column. So what I want to do now then is tell you how do we put this DOC processing in the context, or how does it fit in within the global carbon cycle? And that's what I've got here. So I'm going to walk you through the modern active global carbon cycle, and shown here in petagrams of carbon for pools and petagrams of carbon per year for fluxes. So starting looking at between the atmosphere and land, the net of photosynthesis and respiration is about 3 petagrams of carbon per year uptake by land. And of course, we have what's driving climate change is human emissions, both from fossil fuels and land use change, return of carbon to the atmosphere at a flux of about 9 petagrams of carbon per year. The oceans take up about 2.2 petagrams of carbon per year. So what I want to draw your attention to is if you look at the values, again, the 3 and the 2.2 here, these are comparable, the same order of magnitude as the flux of carbon from land through inland waters or through fresh waters, right? This 2.7 and 0.7, where much of this carbon is dissolved organic carbon. Right? Now, some of that dissolved organic carbon is exported through rivers to the ocean, and some of it is returned to the atmosphere as carbon dioxide, which is shown as this evasion flux of 1.2 petagrams carbon per year. What we've contributed to this diagram here is that of that CO2 that is evaded to the atmosphere, there is biological component, there's respiration of DOC, again, the more conventionally measured, but that sunlight oxidation of DOC to CO2 is also an important source contributing to that evasion term here. So since we've learned that, there are 2 knowledge gaps that my group has been consumed with the past few years that I'm going to tell you about today. And the first one is, now that we know that this photochemical oxidation of DOC to CO2 is important here within the global carbon cycle, what are the controls on that process? So in inland waters, what controls that DOC oxidation to CO2 by sunlight? And the second question is, will this processing of DOC in inland waters change as more of that DOC may come from permafrost soils? So I think probably many of you are all aware that the Arctic is warming twice as fast as the rest of the Earth, causing permafrost soils to warm and thaw. That's a concern because Arctic and boreal soils contain about twice as much carbon as is currently in the atmosphere. Right, this carbon has been frozen. It's not been participating in the modern carbon cycle for thousands of years. And so what we're concerned with is the feedback here. So the extent to which this carbon is returned to the atmosphere as either carbon dioxide or methane can then further be a positive feedback, furthering climate change. And that's called one of the ways in which warming in the Arctic may amplify climate change, or the Arctic amplification of climate change. Right. So that's an overview. And now, so my specific research questions and answers here, starting with the first one, is again, why, what are the controls on this sunlight oxidation of DOC to CO2? Why do we observe more CO2 from some waters than others? And I'll tell you now that the answer is iron, and we think it's iron in the water that catalyzes an important reaction. Then the second question is, as permafrost soils thaw and more of the DOC that is exported from land to freshwaters is that older permafrost DOC, will sunlight be able to oxidize that DOC? And the short answer is yes. We found that sunlight can oxidize old permafrost DOC, and enough that it matters for Arctic amplification of climate change. So that's it. That's what so the rest of my talk, I'm going to try to convince you of these answers by telling you how we arrived at them. Right, starting with my first question here. So we've been measuring rates of DOC processing, or each major pathway of DOC degradation or processing by sunlight and microbes in the water from many lakes and streams over 3 annual cycles in this initial study that we did, the summers of 2011, 2012, and 2013. Right, in the Alaskan Arctic, so this is Alaska here. This black box denotes or shows you on the map where all of our samples came from. Again, this is above the Arctic Circle. It's in the north slope of Alaska, which is what you see when you zoom in here. That's the black box zoomed in. We didn't sample 135 different lakes. Rather, we sampled about 20 to 30 lakes many times over these 3 summers. Likewise, we sampled several streams and rivers, including the Kuparuk River, which is a major river draining the north slope of Alaska. And several times throughout the summer, every summer, we sampled the Kuparuk River at these different sites, from its headwaters in the Brooks Range all the way to its mouth at the Arctic Ocean. And what we found by studying these waters repeatedly over time is that some of them, the DOC in some of them is really good at producing CO2, whereas we got less CO2 from the DOC in other waters. And that's what you're looking at here. On the y-axis, we have the aerial water column rate of photomineralization in millimoles of carbon dioxide per meter squared per day, shown as the average from different sites or groups of sites that we studied. So here's Naviat Creek. This is a small headwater stream near Tulip Field Station, our base operations in the Alaskan Arctic, as well as the average for coastal plain lakes, which is a group of lakes that we sampled on the coastal plain. All of the sites, the average of all the sites in the Kuparuk River, the average of Tulip Lake, and the average for one of the major streams that drains into Tulip Lake. So what we observe is that the rate of CO2 from photomineralization of DOC varies by about 8-fold across these different streams, rivers, and lakes. All right. And so to understand why, what I need to tell you about now is that the water column rate of photomineralization, what you're looking at on the y-axis here, depends on the product of 3 spectra, or 3 wavelength dependent terms. So what I mean by spectra is something that depends on wavelength, and the first of these 3 spectra is the photon flux, or this is what we get from sunlight in mole photon per meter squared per day, again as a function of wavelength on the x-axis. The second spectrum is the absorption spectrum by the DOC, or by the chromophoric light absorbing fraction of DOC called CDOM. How much DOC is there in the water column to absorb light at all these wavelengths. And then finally, the third spectrum is the apparent quantum yield spectrum. So that's just the yield of, in this case, carbon dioxide produced per mole photon absorbed by DOC, again, as a function of wavelength. So if you multiply these 3 spectra together, what you get is this action spectrum here. That's the water column rate of photomineralization in micromole carbon dioxide per meter squared per day, again, as a function of wavelength. And if you take the area under the curve, or you sum up all these values, you would get the total daily rate of photomineralization in the water column. So these first 2 spectra, they're relatively well constrained because they're fairly easily measured. So we have pyranometers that measure the UV and PAR at Tulik Field Station all summer. And we also have thousands of measurements of the DOC absorption spectrum. So for example, this starting over here on the left, you can see this aerial view of the landscape in the Arctic. It's covered by water, lots of shallow ponds and streams and lakes. And in general, these waters contain high concentrations of sunlight absorbing DOC. This is my recently graduated PhD student, Adriana Trusiak, holding up some water here. You can see that it's very colored, very good at absorbing sunlight. And we measure that absorption spectrum in the field and have lots of measurements there. So it's this last spectrum, this apparent quantum yield spectrum, that is the most difficult to measure and we know the least about. And photomineralization rates often depend most strongly on this apparent quantum yield spectrum. So again, the apparent quantum yield is just the mole product. In this example, the product is carbon dioxide produced per mole photons absorbed by the reactant, where in this case, the reactant is the light absorbing fraction of dissolved organic carbon. And again, at every wavelength, involved in the reaction. So we're interested in from the UV out to the visible. And again, because this rate of photomineralization is so sensitive to the apparent quantum yield spectrum, we've spent a lot of time testing hypotheses on what controls the magnitude of the apparent quantum yield spectrum. And what we found is that it's iron. So what I have plotted here on the y-axis is the photomineralization apparent quantum yield in millimoles of carbon dioxide per mole photon, but just at 350 nanometers here. So we can look at the magnitude at 1 wavelength. And again, it's the average photomineralization apparent quantum yield measured over time at our groups of sites or different sites in the Alaskan Arctic, plotted versus the average dissolved iron in the water column of these different streams, rivers, and lakes. And what we observe is that the photomineralization yield of carbon dioxide increases with increasing iron concentration. So why is that? And our hypothesis is that iron is good at catalyzing the oxidation of organic acids within the DOC pool. And that's what's shown in this conceptual diagram here. So DOC contains many organic acids. Just an example of an organic acid you might be familiar with would be acetic acid and vinegar. The organic acids in DOC are probably a lot more bigger and complex than that, but that's just to give you a frame of reference there. The organic acid is represented here as a carboxyl group, abbreviated as the COO minus. And we think that this organic acid, this carboxyl group, is in a chemical complex with ferric iron. And that complex is good at absorbing light. And in the process of absorbing light, the ferric iron is reduced to ferrous iron. The carbon in the organic acid is oxidized to carbon dioxide. And in these oxic surface waters, the ferrous iron can be oxidized back to, or the ferrous iron can be oxidized back to ferric iron, and that cycle may continue. I showed you in the past slide here this relationship with iron because it's the simplest piece of evidence I can show you without getting into the nitty-gritty details of the DOC chemistry. But we do have lots of supporting evidence for every piece of this conceptual diagram that I'm showing here that I'm happy to explain if anyone is interested. So this is what we think is really a key control on the CO2 that we get when DOC is exposed to sunlight. So if we've got iron in the water, we get more CO2 because we think this reaction is happening. And if we have less iron, we get less CO2. Right. So that's the first question. The second question is, how might this change as more of the DOC that is entering surface waters is coming from the deeper permafrost soil layer? Right. And again, with the Arctic warming, temperatures in the permafrost are increasing, or they have been increasing over time. As shown here for these temperatures in these deep boreholes, this is a record from different sites in the Alaskan Arctic going back to when the measurements started to be made in the 1970s through about 2016. And over time, the temperatures, soil temperatures are increasing. And in addition, the surface of the permafrost is thawing, meaning that there's a deepening of the active layer in the soil. Right? Where that active layer of the soil is just the layer of the soil that thaws out every summer in the Arctic. And that's, again, the top 30 centimeters or so. Whereas below that, you have lots of organic carbon in the permafrost, which has been frozen for thousands of years. So this is carbon here that has not been, not had water flowing in it, the carbon hasn't been participating in the carbon cycle. And remember that permafrost soils store tremendous amounts of carbon. And that because this carbon has been frozen for a long time, the organic carbon in these soils can be old, up to 45,000 years old in some locations. Right? But as the active layer deepens and starts to get into that permafrost layer, then water can move through the soil and bring that older permafrost dissolved organic carbon into surface waters. However, permafrost DOC can also get into surface waters in a more dramatic fashion. And that's when ice-rich soils thaw, they destabilize and form these kind of slow-moving landslides. And it's called thermocarce failures, and that's what you're looking at here in this photo. Thermocarce failures tend to form on rivers and lakes, such as this one that we're studying here. This is a photo that I took. And what you're looking at here is, you can see this mud, and that's just, permafrost organic carbon that is then dissolving into the water, the lake water here, where it can be exposed to sunlight, or where it will be exposed to sunlight. And there has been a debate about whether that sunlight can oxidize permafrost DOC to CO2. Some have argued yes, and some have argued no. And we thought the only way to definitively settle that debate is to measure the radiocarbon age of the CO2 produced when the DOC is exposed to sunlight. So our hypothesis was that the CO2 produced from photomineralization of DOC is as old as the CO2 from which it came. Right. So if we were to measure the radiocarbon age of the DOC here on the y-axis, as well as the radiocarbon age of the CO2 produced when that DOC is exposed to sunlight, our hypothesis is that the data would fall on this 1-to-1 line, and that is generally what we observed. So again, we here on the x-axis, we've got the delta 14C of the DOC before sunlight exposure. And then on the y-axis here, we've got the delta 14C of the CO2 produced from the DOC. And that difference in the delta 14C between the DOC and the CO2 was about 2 to 16%, or at most 16%. That's why you see that most of our permafrost DOC plot pretty close to that 1-to-1 line, meaning that in general, there was a pretty small difference in the delta 14C of the DOC and of the CO2 produced when that DOC is exposed to sunlight, which supports the hypothesis that the CO2 from photomineralization is as old as the DOC from which it came, which for our samples in the Alaskan Arctic ranged from 4,000 to 6,000 years old. The second point I want to make here is that we made these measurements for DOC exposed to UV, light in the UVB, and light in the UVA, which tells us that even longer wavelength, less energetic, UVA, approaching visible light can oxidize old permafrost DOC to CO2. And that's important because it gives us confidence in the shape and magnitude of the AQY spectrum for permafrost DOC, which is what is plotted here. Again, we've got the photomineralization AQY in millimoles of carbon per mole photon absorbed on the y-axis. Again, it's a spectrum, so versus wavelength. The points are all the different individual wavelengths where we made these measurements for different permafrost DOC leachates. And just like we observed in the surface waters, well, I should say first, of course, permafrost DOC was oxidized to CO2 at all wavelengths that we studied. And then just as we observed for surface waters, there was a range in magnitude. So any wavelength you look at, you can see that some permafrost DOC has a lower yield of CO2 and some has a higher yield of CO2. And just like we observed with surface waters, that magnitude and yield seems to be controlled by iron. So here we've got plotted at just one wavelength, 350 nanometers, the photomineralization apparent quantum yield versus the total dissolved iron in the permafrost DOC leachate. And in general, there's a pretty nice correlation. The photomineralization yield increases with increasing concentration of dissolved iron, with the exception of one outlier, which we think we can explain, and I'm happy to if anyone is interested in that. So if we take the average of all these permafrost DOC, we can see that it lines up pretty well with the surface water points here, supporting the hypothesis that where we have more dissolved iron, we get a higher photomineralization yield, or more CO2 produced when that DOC is exposed to sunlight compared to waters that have less iron. Again, why do we think that is? We think that iron is catalyzing the oxidation of organic acids into carbon dioxide. And we have amassed a lot of evidence that this iron, this photodecarboxylation is an important source of CO2, both for DOC that is draining from the active layer, as well as DOC draining from the deeper permafrost soil layer. So, however, there is one important difference between the DOC draining from the active layer and the deeper, older permafrost DOC, and that is in the shape of the apparent quantum yield spectrum. So I'm plotting the apparent quantum yield spectrum again here. the yield of CO2 per mole photon absorbed by the DOC versus wavelength. The difference here is that I'm plotting it, the y-axis in log space here, to emphasize the difference in the visible, or UVA into the visible portion of the spectrum. What I've got plotted is the average and 95% confidence interval for 3 surface water DOC that we've studied: Imnaviet Creek, Kuparik River, Tuluc Lake, and then the average of all of our permafrost DOC. The takeaway point here is that the permafrost DOC has significantly higher yield for CO2 production into the UVA and visible portion of the spectrum. And that's really important for water column rates of photomineralization because there's a lot more visible photons reaching the water than UV. So remember that the rate of photomineralization in the water column depends on the product of spectra. I'm showing you 2 here. I'm not showing the CDOM absorption spectrum. I'm showing these other 2, which is the apparent quantum yield. Again, not in log space this time, but you can still see the difference in the UVA into the visible portion of the spectrum here. For the black line is the surface water DOC in Imnaviet Creek. This blue line, or turquoise, is permafrost DOC in the same watershed, having significantly higher yield in the UVA into the visible there. And that yield then is multiplied by the photon flux, right, where we have a lot more photons in the visible, about 10 times more than in the UV. So if you multiply these together to get the rate of photomineralization, then what you can see, if you were to take the area under the curve here, the blue curve is for the permafrost DOC, the black line is for the surface water DOC. If you integrate under the curve, then you get about double the rate of photomineralization from the permafrost if the water column were entirely made up of the permafrost DOC versus the surface water DOC. So again, this small difference in, or what looks like a small difference in the UVA into the visible can give you double the CO2 because there's just a lot more visible photon flux. Right, so the shape there is really important. Right, so what if the water column isn't all permafrost DOC, which it certainly isn't now? And so we did that calculation. So what's plotted here on the y-axis is the percent increase in the rate of carbon dioxide produced from photomineralization of DOC as a function of how much of the DOC in the water column is permafrost, starting with 0. So this is our current day condition, you know, going up to 100% permafrost DOC. And we did this for a range of scenarios, different streams, lakes, that we studied, you know, mixed in with the different permafrost DOC that we studied. And on average, there's an increase in the rate of CO2 produced, and that's due to that higher magnitude of the AQI as well as the shape of that AQI spectrum. So that, you know, as the water column as the DOC in the water column is comprised more permafrost DOC, we could get up to about a 75% increase in the rate of CO2 produced. So, so to understand then what the implications are for the future, let's look at the Arctic carbon budget, both now and how it's predicted to change in the future. So starting with the terrestrial balance, the net ecosystem exchange ranges from plus to minus 10 grams of carbon per meter squared per year. So that's, you know, roughly in balance here on land. And right, whereas the aquatic loss of carbon, that's dissolved organic carbon, dissolved inorganic carbon, and particular organic carbon to the atmosphere and to the ocean, ranges from 4 to 6 grams of carbon per meter squared per year. Right. The important point here is that in a year where that net ecosystem exchange is 0, so no net or imbalance, then all of the loss is due to what's happening in freshwaters. It's the aquatic loss that's important. So the aquatic loss could be 40 to 100% of that long-term mean net ecosystem exchange. Right, so that's current understanding, pointing out again this importance of what happens to carbon in the water on the landscape. Dave McGuire and others have modeled what may happen in the future under a range of scenarios, and concluded that on average, you get about 208 petagrams of carbon lost to the atmosphere by the year 2300, or between 2010 and 2299. However, their predictions here, this loss of carbon, does not include the photomineralization of DOC to carbon dioxide. So we took what we have learned from our work, and we estimated how much that this predicted 200 petagrams carbon loss may be off by not including the photomineralization of DOC. We concluded that it's too low by about 14%. Right. And we did that again by taking the rate of photomineralization now and making some assumptions about how much of the land surface would be surface water, and then allowing the percent of the DOC in the water column to be increasingly permafrost starting from now up to the year 2299. So we would argue that that's an important contribution, or important underestimate, and our estimate here that they're too low, these predicted 208 petagrams carbon loss is too low by 14%, is a conservative estimate for several reasons, two of which I've highlighted here. So one, it doesn't consider the other ways in which sunlight can turn permafrost DOC into CO2. For example, that can happen in coastal waters or in the ocean, which we didn't include in our estimate. Another process that's important is that the DOC that's left behind after sunlight after it's been exposed to sunlight is often more labile to microbes. So microbes can respire more of it compared to the same DOC kept in the dark. And so we did not include that part in our estimate here. In addition, we know that ice out is happening on lakes earlier, and that's exposing then more surface waters to sunlight at a key time of year. And that's when the sun angle is highest in the Arctic, and so the photon flux is the greatest. And we did not include that any changes there in our estimate. All right, so in conclusion, I'll just return again to my question and answer here. Why is there more CO2 from some waters than others? Well, we think we understand why, and we think it's that iron plays a key role by catalyzing a specific reaction that turns dissolved organic carbon into CO2 with the help of sunlight. And will this process be important in the Arctic? And our answer is yes. Sunlight can oxidize old permafrost DOC to carbon dioxide, and enough so that it contributes to Arctic amplification of climate change. Right, and with that, I'm going to end here and am happy to take any questions.
Thank you very much. I can't see everyone on the screen. I'm going to try to.
Yeah, me neither.
Okay, if you can raise your hand electronically. Okay, wonderful. Lawrence.
Hi, Professor Cory. That was a wonderful talk. Thank you. I had a quick question about like your second to last slide, basically that 30 petagrams per carbon, 30 petagrams of carbon by 2299.
Yeah.
Do you think that thaw lake drainage or increased aridity in the Arctic in the next 200 years? Sorry to ask that kind of question. I guess in the next 300 years, do you think increasing increasing drainage of thaw lakes might dampen that effect a little bit? But I know that that's not a there's no good quantification of that. So I don't want to.
Well, I mean, so yeah, I think it's a good question. I think Claudia asked me that question indirectly beforehand, and I think what I started thinking about when Claudia asked me that question is that you could take this estimate here and just say, well, how much less would you have, if you lost these lakes? So, as I'm sure you know, some studies predict more water, some predict drying and less lakes. We did not, and because it's not at all certain, we didn't allow the percent of the land surface that is water to change. We just kept that constant. But I think you could take our estimates and play with them in all kinds of ways. And I'm looking sideways because I have the Zoom part over there. So yeah, you could, I think you could, you could simply say it could range between 0 and 30 petagrams of carbon. Maybe a little more complicated than that, but.
Okay, thank you.
François and then Sergey.
Hi, it was a nice seminar. Do you have an explanation or a hypothesis why the type of organic carbon that's in the permafrost is different than the stuff that is making it the modern cycling stuff? Like, why is there preferential storage of carbon that can absorb longer wavelengths? Is there a reason?
Yes, yeah. So, okay. So first of all, so there's a few reasons. One of them may be methodological. So let me see here if I can, I'm going to try to find more quickly the right slide. Okay, so first, here's the methodological reason. My former PhD student, Colin Ward, who's now his own independent, amazing scientist at Woods Hole, he came up with this brilliant way to measure the apparent quantum yield using LEDs. And his paper on this just came out, and this is transformative. It's a game changer. Because he worked on permafrost DOC for his PhD with me, he was really eager to let us test this on permafrost DOC, which we did. And so these measurements that I've shown you are the first wavelength specific measurements that anyone has made on permafrost DOC. There have been actual wavelength specific AQYs measured, but it's not really very many overall. And so the way that we measured that spectrum from surface waters was by broadband exposure to sunlight. And then what you have to do is assume, assume a spectrum. So in another way of saying that is that basically there's no other result that we could get, because we assume that that the surface water has this exponentially decaying spectrum based on the very few measurements that have been done. So what we may find is that actually the surface water DOC has a shallower slope than we have assumed as well, and that is the first thing that we are testing when we get back in the field this summer. So we've made our own LED system, and I'm about to ship it to the Arctic. So short answer, the first part is it may be a method issue. The second part is that, yeah, it could be a chemistry issue or real chemistry here. So one of the things that we found, was when we measure the loss of carboxyl carbon by C13 NMR, we find that for permafrost DOC, it can account for about 90% of the CO2 produced. Whereas for DOC draining from the active layer, it accounts for about 30 to 40%. So that would indicate that there's maybe something about the partnership between iron and the carboxyl carbon, or maybe about that carboxyl carbon independent of iron that is just more reactive, and that we don't understand why. But the whole kinds of, but I guess the simple way of saying it is that this photodecarboxylation appears to be very important for permafrost DOC, whereas for the DOC draining from the active layer, I think there's other reactions that are happening as well.
Great questions. Sergey, can you unmute yourself and ask the question?
Yes, sure. Can you hear
Thank
me?
you.
Yeah.
Yeah. I was impressed that your quantum yield remains high at long wavelengths, which is, to me, suggests maybe involvement of triplet state chemistry, not just ion. Is this a possibility you considered?
We haven't, that is one thing that we haven't considered. And I don't even know if, so my postdoc advisor, Chris McNeil, I mean, he's done much more work on triplet state photochemistry, but I don't even know if I can't think of any wavelength dependent data for DOC on that off the top of my head. There may be some older paper that I'm thinking, but I'm not sure. What we have looked at, we've looked at Fenton chemistry, hydroxyl radical production, and that can't account for everything that we're observing, but I am always eager to consider other pathways. So, you know, if you have any ideas, I'd be happy to hear them.
No, I found your quantum yield measurements impressive and inspiring. So, and also a note, so we study what's called brown carbon in the atmosphere, and we find that power law fits the absorption profile better than exponential decay. And I think it's also the case for dissolved organic matter. I mean, they're not the same chemically, but we usually use dissolved organic matter as a standard.
Oh, wow.
Measurements. So, and I think the power law is a better representation, but it probably creates only minor adjustments to your analysis.
Yeah, I mean, there's probably in your field as in mine, there's a lot of disagreement about the fitting, and it can, yeah, and overall, I'd say it doesn't change the answer, but there are some cases where those small differences can have a big impact.
Okay, and we have a question from Michael Prada.
Hello, thank you, very nice. Sort of two simple ones. Isn't this happening in the land surface, slightly wetted all over land, not just in water columns? Why would you expect this to be limited to water columns as opposed to just the land surface? And the other minor one is that your quantum yield will depend on what else is absorbing in the water column, I would think. And
Yeah.
so the question is, how do you separate out other absorbers which are not producing anything, but are just interfering with the rate at which your photolysis is happening?
Yeah, both are great questions. So let me start with the second one because I can answer that a little bit more easily, although I'm not sure if I have the data in here. I won't spend time looking for it. But so we actually, we measure light extinction profiles in the field using a compact optical profiling instrument, which is made for the oceans. It's a little bit harder to deploy it in a pond. But so we measure the light that's being absorbed in the water column, and then we compare that to the absorption profile that we measured from filtered water in the laboratory on a UV-Vis spectrophotometer. And they agree, like the data plot, basically on a 1-to-1 line for any wavelengths from the UV into the visible. So what we and many others have concluded in fresh waters, particularly high DOC waters, that DOC is the main light absorbing constituent in the water. Of course, there are certain systems where that's not the case. Like I also work on Lake Erie, which is plagued by harmful algal blooms and has relatively lower DOC and lots of biomass. And in a water column like that, you actually have to consider that other things are absorbing in the visible. But even in Lake Erie, mucky green pea soup Lake Erie, in the UV, DOC is still absorbing 50% of the light in the summer. So in the Arctic, again, we have, even though there's high iron, DOC is absorbing most of the light. And I can show you that, like here, if we have a picture, right? You can only see iron in the water where you have low DOC concentrations, because the iron precipitates out, and that's that red color, right? So where there is high DOC, you get high dissolved iron. We don't have other things that would absorb light, like high nutrients or really anything else. So that's how we're very confident in the DOC. Again, compared measurements in the field, but also having a good understanding of what's in the water column. And lots of people have done that work, not just my group. And then your first question, how do we know, why are we looking at just the water? Well, so for one thing is, the waters in the Arctic are mostly unshaded, so that makes it easy. You don't have to deal with shading by biomass or shrubs or the kinds of things Claudia would consider. But not very many people at all have looked at photodegradation of leaf litter or plant matter. But there are some groups, including I think Jennifer King in California, I'm forgetting what university she's at, and also Amy Austin in Argentina. They're starting to look at that, and my understanding of, or they have been for a while, my understanding of their work is that that's photodegradation of leaf litter is important basically where in very arid systems, and where you need water is limiting, I think, the respiration. So I don't know, it could be important in the Arctic, but we've never looked at it, and I'm a little bit intimidated to do so. I think it would be much harder. Those are great questions.
Yeah, if you have more questions in the audience, please raise your electronic hand. I have also a question. How well do we know the flux of iron in the Arctic rivers, and what controls that? And the second question was that when you say permafrost DOC, what depth does that come from? Because soil profiles can be quite complex.
Yeah, right, great. First question, let's see, yeah, for iron. So here's one. This is work that my PhD student Adriana did. So we're also looking at some, well, it doesn't matter. The point here is that one of the controls on iron export from soils to surface waters is just DOC concentration. They're tightly coupled. That's probably, well, it's, you probably know, Claudia, when you have these soils are waterlogged, and so they get very anoxic. You get reduced iron buildup, which is dissolved, and then that gets flushed into surface waters. Some of that iron, we think, of course, is complex with the DOC, which is one reason why there's a strong relationship. And what I should have pointed out is that these are for different sites in the Arctic. These are averages for different sites. So, the iron and the DOC co-vary on the landscape, and that's what we see certainly in the surface waters, but then also the export of them from the soils co-vary. So again, these waterlogged soils are good, building up both iron and DOC, and then that gets flushed to surface waters. For your second question, what depth? So for someone like you, we probably did not go very deep. I think we were, we were definitely into the permafrost, but within the top 1-2 meters. So not very deep. And it is a good question of whether this answer would change if we were to look deeper. And all I can say now is that from what our relationship between that radiocarbon age of the CO2 produced and of the DOC, radiocarbon age of the DOC, we have that nice relationship. I would say at this point, which is not with a lot of samples, but at least with what we know, we'd expect that we should at least get some CO2 from DOC.
Okay, thank you. And then there's another question by Francois.
Sorry for asking two questions. Does this have any relevance to perhaps to iron fertilization in the ocean? Could we get more DOC photo oxidation in addition to fixation, do you think?
That's a great question, and I want to just defer right to Ellen on that one, but I'll try to. I don't know, because the pH is so different, the salt content is so different. So, you know, and of course the iron concentration is really, really different. Well, right, but then you'd fertilize, you'd add iron. I'm not sure that. So one of the things that we don't know is like how much of this really reactive carbon gets oxidized or removed before it gets to the ocean, right? And this reactive carbon is the carbon that's good at forming that chemical partnership with the iron. So it may not matter if it's gone before it gets there, or maybe it's gone in the coastal ocean. So that may be one key factor. But then also, I think, you know, you have competition perhaps for complexing with those acids with the salts, and you know, with I don't know. So it'd be fun to think about. And
Thank you.
Okay, well, thank you very much for a great talk and really lively discussion today.
thank you, great questions. I really appreciate it.